different types of stem cells. There's embryonic stem cells and then there's adult stem cells. And basically you take cells and you can use the genes from them, something like that, in order to help fight diseases and stuff. A lot of genetic modification kind of thing. That's really all I know about it.
I donated some when I had my first child. Thought it sounded like a good thing to do.
It's good because we learned about, last year in biology, we learned that stem cells are good because you can use them to like build other things.
And that's all I know. That's it.
The stem cells that we work with primarily come from embryos, and they're very early stage embryos, so just a couple cells. And these were originally discovered in mice, and so people first found that these embryonic stem cells, we'll call them, could be derived from the embryos of mice. And so just when the embryos are a few days old or just a day or two old, they have cells that can contribute to all the different tissues of the body. There are cells in your blood that that are called stem cells, and they make new blood cells throughout your life. That's the basis that allows things like bone marrow transplants that go on over at the Fred Hutch to work, because you really transplant a cell that can keep making new cells over and over and over again. So your bone marrow transplant has to include those stem cells in order to be able to fully repopulate your blood cells again. So these are examples of adult stem cells. So there are adult stem cells and there are embryonic stem cells. There's an alternative to that has fewer objections. About 10 years ago, a group in Japan discovered a way to convert any cell in your body, potentially, into a stem cell that's very much like an embryonic stem cell. And that's called an induced pluripotent cell, or iPS cell. And they're very much, the induced pluripotent stem cells are very much like the embryonic stem cells. And so we sometimes use those in our studies as well. They behave in most assays just exactly like the embryonic stem cells, so I think that in many cases they can replace the embryonic stem cells. And so they don't require that any new embryos be destroyed in order for those cells to be generated.
Kind of just gives you an idea of what we're working with and where it's coming from and where is the taboo that happens with stem cells. And so obviously the biggest issue is that they do come from a human embryo. It involves the destruction of a human embryo, which a lot of people find very upsetting. And so one of our favorite slides to show is this one here. It's a plastic model of human development, just giving you an idea of exactly how small it is that we're working with. You can't even see it on this scale with the naked eye. It's this invisible space right here. And so if it were in a human being, which these never are because they're made in reproductive facilities, they would not have been planted into the uterus yet, and that person would not be considered pregnant. It's that early on, 5 days post-fertilization. Very, very, very early on. And so where we get them, because we run off of private funding, we're one of very few labs in the country that can make stem cell lines, can accept these donations from reproductive facilities.
At least some of these embryos would have had that potential, so the fact that you're destroying them, even at a very early stage like this, where there's only, I don't know, maybe less than 50 cells or something in the embryo at that point, still there's ethical objections to that. Some people regard that as almost like you're aborting the fetus. Even though it's a much earlier stage than abortion, it's still considered by some people to have been a human life that was destroyed. And so for religious reasons, they wouldn't want to use embryonic stem cells.
These are the incubators that we keep the cells in. They're at 37 degrees Celsius and what we would call normal oxygen concentration, which is around 20%. So we're basically just trying to mimic human body conditions in here. So you see a couple things here. This is a colony of human embryonic stem cells. probably a couple hundred of them, and they grow in these colonies. They like to stick together, and they like to be a certain size and a certain density. And all of this stringy stuff that we have in the background here is, they're called mouse embryonic fibroblasts, and they're basically kind of like, a fibroblast is kind of like a skin cell, basically, kind of a matrix. And we put them on the plates before we put the cells on them to get the plates to stick down, because they need something to adhere to. Otherwise, they're kind of all alone, and just like, stem cells get stressed out really, really easily. I'm part of the heart regeneration program here at the University of Washington. Basically what that is a preclinical program for transplanting human cardiomyocytes into the hearts of patients that have had heart attacks. And so we've done this now in rat and guinea pig and monkey, which means that we're ready to go to human. And basically what it involves is taking stem cells like we saw before. We mimic human development by kind of spiking the food with different proteins and chemical messengers that are around during fetal development. And within about 2 weeks, actually technically it was only 8 days, this one's that we're going to look at. It's only been 8 days since they were what we just saw, the stem cells. And you've got immature eating human cardiomyocytes. These are the stars of the show here. So, if I can get them to be focused a little better there. So we didn't have to do, aside from spiking the food with, the right chemical messages, we didn't have to do anything to tell these guys to start beating. There was never a time when we shocked them with electricity or had any sorts of outside manipulation. I just came in this morning, actually. It was the first day they started beating. I just came in this morning, and they're beating away. So we're looking at a single one of these wells here. I would say there's probably about 1 million cardiomyocytes in one of these wells, and we need 1 billion for a single injection, for a single human.
They could be used to treat any disease where people have lost one of their functions through the loss of a cell that doesn't have intrinsic stem cell.
Using iPS cells to make those 6 cells in a dish so that we can do whatever we want with them, and the patient doesn't have to go through side effects, doesn't have to go through painful procedures to try to find the right drugs for them. So I think that's probably the most exciting application for iPS cells today. Obviously in the long term we would love to be able to do what we're doing with stem cells with iPS cells so that we don't have to immunosuppress, so that we're not putting foreign cells into somebody's body. We can literally take a sample from you and put it right back in as heart cells, or as pancreatic islet cells if you're diabetic, or retinal cells if you're going blind. And so that in the long run is really, really cool to think about, but like I said, it's a long, long, long run.
The immediate ones will be treatment of diseases where a specific cell is lost. But I think ultimately you can see that this could be used to actually, it'll treat diseases that aren't so serious in the future. So I think that initially we'll just treat diseases that have no other treatment. But eventually we might treat diseases that the drug treatments just aren't perfect or aren't ideal. And so eventually we'll treat more and more things with the stem cells. That's my guess.